Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Today, we're looking at something pretty fascinating, a study that might just flip our whole understanding of PTSD. For years, right, we've mostly thought of post-traumatic stress disorder as sort of a broad issue, like an imbalance in brain chemistry, generally speaking. But this big new study in Nature says, hold on, it's much more specific. It's framing PTSD as a complex, cell-type specific regulatory disorder.
Yeah, that's the key phrase, cell type specific. This isn't just, tweaking the existing picture. It's really a fundamental shift.
A paradigm shift, even.
I'd say so. It changes how we even think about mapping trauma's effects on the brain, right down to the individual cells involved. And that's huge because it reveals these very specific vulnerabilities, potential new targets for treatment we just hadn't seen before.
Okay, so let's back up a bit. Why is this kind of deep dive so crucial? PTSD affects so many people.
Billions, yeah.
But figuring out which specific brain cells are actually changed by trauma, that's been incredibly tough, hasn't it?
Oh, absolutely. A huge challenge. A lot of the research relied on, well, they call it bulk tissue analysis.
Which is kind of like...
Imagine trying to understand a symphony, a really complex piece of music, by just hearing all the instruments mushed together into one sound.
Right. You know something's playing, but you can't pick out the violins or the trumpets individually.
Exactly. Or researchers might look at peripheral blood, which is even further removed from the brain itself. And that limitation, it really matters. Without knowing which cell populations are involved, this cell-type resolved map, they talk about treatments stay broad.
Like using a shotgun when you need a scalpel.
Precisely. If we knew which neurons, or maybe which glial cells, are misfiring and why, that's the key to developing truly targeted therapies. Think new drugs designed for specific cell types, or maybe even neuromodulation focused on particular circuits. This study is trying to build that essential map.
Okay, so let's unpack the study itself. Cool. Because the scale is just, wow.
It's massive.
They didn't just analyze a few hundred cells. They profiled, 2.1 million individual cell nuclei?
2.1 million. From a really critical brain region to the dorsolateral prefrontal cortex.
That's involved in decision-making, regulating emotions, sort of the brain's executive control center.
You got it. And it wasn't just a few brands. They looked across 111 human post-mortem brain samples.
From people with PTSD, people with major depressive disorder, that's important as a control, and healthy controls too.
Yeah, the MDD group is key for comparison.
So a huge data set. How did they even manage to analyze that many individual nuclei in such detail?
Well, they used this really sophisticated multi-omic approach. So not just looking at one thing per cell, they used single-nucleus RNA sequencing, or snRNA-seq.
Okay, to see which genes are turned on, which are being expressed.
Right, but they also combined it with single-nucleus ATAC sequencing, SNAC-seq.
Which tells you.
That shows which parts of the DNA are open or accessible, basically which genes could be turned on because the machinery can get to them. And in some cases, they even did both from the same nucleus using something called SNAN multiome. That gives incredible resolution.
Wow. So you know not just what the cell is doing, gene expression, but also what it's poised to do, chromatin accessibility.
Exactly. It's like having the sheet music and hearing the instrument play.
So they built this incredibly detailed map. What was the first big finding that jumped out from all that data, like the headline from Figure 1?
Well, the first really striking thing, looking at overall cell populations based on gene expression, was a specific change. They looked at all the main cell types neurons, here,
Like astrocytes, microglia.
oligodendrocytes, endothelial cells, yep. And they found a significant decrease specifically in oligodendrocytes, the OLGs, in the PTSD group.
Compared to both the healthy controls and the MDD group.
That's right. So it looked specific to PTSD in this comparison. And beyond the cell numbers, they found over 1,000 genes whose expression changed in PTSD. But here's the kicker. No single gene was messed up across all the cell types.
Reinforcing that cell type specific idea.
Precisely. It's not one broken gene everywhere. It's different genes acting up in different cells.
Can you give us an example, one gene change that was particularly surprising or revealing?
Sure. There's a gene called FKBP5. Now, researchers have known for a while it's involved in the stress response.
Right, often studied in neurons.
Exactly. But here they found it was significantly upregulated, turned on more in endothelial cells.
The cells lining blood vessels in the brain.
Yes. And also in oligodendrocytes. The endothelial finding was particularly unexpected. It points towards maybe a neurovascular component to PTSD involving FKBP5, something we hadn't really appreciated.
So stress isn't just hitting the neurons directly, but maybe also the brain's plumbing, the blood supply.
It certainly suggests that pathway might be involved, yeah. A vulnerability in the blood vessel lining itself.
And what about that comparison with major depressive disorder? You said that was key. How did PTSD look different from MDD at this gene level?
Yeah, this is where it gets super interesting. There was overlap, definitely. About 57–58% of the gene changes were shared between PTSD and MDD, so common biological ground. But crucially, they also found over 500 genes that were altered specifically in PTSD and not MDD in these cell types.
Can you give an example of that divergence?
Sure. A gene like CTNNA3 was downregulated in PTSD neurons, but actually upregulated in MDD neurons.
Opposite directions.
Exactly. Or HSPA1A in endothelial cells showed similar divergent patterns. It really highlights that even though these conditions often occur together and share symptoms, their underlying molecular signatures, cell by cell, can be distinct.
Which has huge implications for treatment, right?
Yeah.
You might need different approaches, even if the symptoms look similar on the surface.
Absolutely. It argues against a one-size-fits-all approach.
Okay, so they found these gene changes in specific cells. But the brain isn't just a soup of cells, it has structure. How did they figure out where these changes were actually happening? Did they, map it onto the brain tissue itself?
They did. And that's where the next piece of tech comes in spatial transcriptomics. Think of it like adding GPS coordinates to the gene expression data.
Putting the microscope on steroids, you said earlier.
Yeah, something like that. They used a platform, 10x Xenium in this case, to map gene expression directly within slices of brain tissue, almost down to the single cell level.
So you can see not just that a gene changed in a certain cell type, but where those cells are located in the brain structure.
Precisely. It lets you validate the single cell findings in their native context and see how these different cell types are arranged and potentially interacting.
And did the spatial map confirm what they saw in the isolated nuclei? Did it line up?
It really did, yeah. The spatial data correlated strongly with those initial single-nucleus findings. Remember that FKBP5 gene being upregulated in endothelial cells?
The blood vessel lining cells, yeah.
The spatial data visually confirmed it. You could see high FKBP5 expression signals clustered right around blood vessels throughout the prefrontal cortex tissue sections.
Wow, so it visually locks in that potential neurovascular role.
Exactly. It makes the finding much more concrete when you see it in situ.
Any other cool examples from the spatial mapping? Something else that stood out?
Well, they also used it to highlight the upregulation of another gene, CXCL14, specifically in inhibitory neurons.
And CXCL14 is?
It's a chemokine, a signaling molecule that's known to potentially dampen GABAergic transmission.
GABA is the main calm-down signal of the brain, right? The main inhibitory neurotransmitter.
That's the one. So seeing CXCL14 specifically pop up in inhibitory neurons in the spatial map, it starts to build a picture of how communication might be getting disrupted. Maybe specific calm-down pathways are being selectively turned down.
Leading to that kind of hyper-excitable state often seen in PTSD.
That's the implication, yes. It provides a spatial anchor for those functional hypotheses.
Okay, so genes are changing in specific cells, in specific locations. The next logical question is, how does this mess up the conversation between the cells?
Right. How does it affect cell-to-cell communication? And that's the next layer they explored, using computational tools like cell chat and neuron chat. These tools basically infer communication pathways based on which signaling molecules and receptors are expressed by different cell types.
Trying to reconstruct the dialogue from the gene expression data.
Sort of, yeah. And they found significant changes in these inferred conversations in the PTSD brains compared to controls.
What was the most striking finding there in terms of broken communication?
One really fascinating thing was about microglia, the brain's immune cells. They found decreased sending patterns from microglia in PTSD.
Decreased sending, so they're quieter.
Apparently so, particularly involving a pathway called SPP1. And what's really interesting is, again, the comparison with MDD. In MDD, that same pathway showed the opposite pattern, increased sending from microglia.
Wow, so maybe the immune response in the PTSD brain is actually suppressed in some ways. That feels counterintuitive.
It does challenge some common assumptions about neuroinflammation in PTSD. It suggests a more complex, maybe even suppressed microglial communication signature, at least via this pathway.
Okay, suppressed microglia communication. What else? Any specific neuronal communication going wrong?
Yes, and this might be one of the most critical findings for understanding symptoms. It involves a specific type of inhibitory neuron, the somatostatin, or SST interneurons.
Okay, SST interneurons. What about them?
The analysis showed significant decreases in their communication output. They seem to be talking less to other neurons, to astrocytes, even to those endothelial cells we mentioned.
So these specific inhibitory cells are becoming less influential.
It appears so. Their outgoing signals are predicted to be weaker.
And what's the downstream effect of that? If SST interneurons go quiet, what happens?
Well, SST interneurons are major players in providing inhibition, largely through GABA. So reduced communication from them directly points to weakened GABAergic signaling. The study specifically found evidence for reduced output affecting key GABA receptors, like GABAer5.
GABAer5, isn't that involved in tonic inhibition, kind of like the background level of calmness in the brain?
Exactly. It contributes to that overall inhibitory tone. So if SST neurons aren't signaling properly to GABAer5 receptors, that background calmness could be reduced.
Which could absolutely contribute to things like hypervigilance, anxiety, the feeling of being constantly on edge in PTSD.
That's the thinking. It provides a potential cellular mechanism for those core symptoms, a failure of specific inhibitory circuits.
Now, this is inferred from human postmortem tissue and computational analysis. Did they try to confirm this SST neuron finding in a living system?
Yes, and this is crucial. They turned to a rodent model of traumatic stress, single prolonged stress, or SPS. In the brains of these stressed mice, they performed electrophysiology experiments and confirmed it. They found reduced GABAergic synaptic transmission specifically from SST interneurons onto other neurons.
Wow.
And they even saw a decrease in that overall tonic GABA current we just talked about, mediated by GABAer5.
So the animal model backs up the human data. These specific inhibitory neurons really do seem to be less effective after trauma.
It provides strong functional validation, linking the molecular findings in humans to actual changes in brain circuit function.
Okay, so we've got gene changes, locations, communication breakdowns. But what's controlling the gene changes in the first place? What are the master switches?
Right, that takes us to the epigenome, specifically looking at chromatin accessibility again, using that ATAC-seq data we mentioned earlier.
The open or closed parts of the DNA.
Exactly. Think of the DNA wrapped around proteins that's chromatin. How tightly it's wrapped determines if a gene can be read. Open chromatin means the gene is accessible. They mapped hundreds of thousands of these open regions, these chromatin peaks. Many of them are in non-coding parts of the DNA, acting as potential regulatory switches, cis-regulatory elements, or CREs.
Like dimmer switches for genes located nearby on the DNA strand.
That's a good analogy, yeah. They control the expression of nearby genes.
So did these switches, these open chromatin regions, actually line up with the genes that were changing in PTSD? Did they connect the dots?
They absolutely did. They found a strong correlation. The differentially expressed genes, especially in cell types like endothelial cells and excitatory neurons, were significantly associated with changes in chromatin accessibility nearby.
So take FKBP5 again. That gene upregulated in endothelial cells.
Right. They found that the accessibility of the chromatin around the FKBP5 gene was highly correlated with its expression, specifically in endothelial and microglial cells.
Suggesting those open regions are indeed acting as switches controlling FKBP5 in those cells.
It strongly suggests it. It helps build that causal chain. Change in chromatin accessibility leads to change in gene expression. It moves beyond just listing genes that change to suggesting how their regulation is altered.
And again, comparing PTSD and MDD, where the patterns of open and closed chromatin, these regulatory landscapes, different.
Yes, significantly so. They identified over 140,000 condition-specific open chromatin regions, or CREs.
Meaning regions that were accessible in PTSD, but maybe not MDD or vice versa.
Exactly. Even if the same gene pathway is ultimately affected in both conditions, the specific way its regulation is altered at the chromatin level can be distinct.
So different switches might be faulty, even if they control lights in the same room, so to speak.
That's a good way to put it. It emphasizes disease-specific mechanisms right down at the level of genome regulation.
Okay, this is building an incredibly detailed picture. Now, how does this all connect back to genetics? To the known genetic risk factors for PTSD that come from huge population studies, the GWAS studies.
This is where they really tied it all together. They integrated the findings, the cell-type specific gene expression and chromatin accessibility, with data from large-scale genome-wide association studies for PTSD.
What did that show? Where does the genetic risk for PTSD seem to live in terms of cell types?
The analysis showed that the genetic variants linked to PTSD risk were significantly enriched within the open chromatin regions, the ATAC peaks, found specifically in excitatory and inhibitory neurons.
So the genetic susceptibility for PTSD largely plays out within neurons, according to this data.
It strongly suggests that these neuronal populations are the primary places where inherited genetic risk variants exert their effects on brain function, ultimately increasing risk for the disorder.
Could they go even further? Could they pinpoint specific genetic variants potentially causing trouble in specific neuron types?
They could, using statistical fine mapping techniques. This allowed them to zoom in on specific DNA variations within those neuron-specific open chromatin regions that are most likely to be the actual causal variants influencing PTSD risk.
Wow. So, for example,
Yeah, well, they looked at a gene region, or locus, called MAD1L1, which GWS studies had previously linked to PTSD. Using their cell-type specific data, they identified a particular genetic variant located in an open chromatin region specific to inhibitory neurons, and this variant was predicted to strongly affect the expression of a neurite gene called ELFN1.
okay, ELFN1. Why is that gene significant here?
ELFN1 is really interesting because it codes for a protein found at synapses, specifically involving interneurons, and its expression was already known to be altered in PTSD studies.
Wait, interneurons. Didn't we just talk about problems with SST interneurons and their communication?
Exactly, and guess where ELFN1 expression is highest? In those very same SST interneurons.
No way. So it all connects.
It seems to. The study proposes this model. An inherited genetic risk variant, near MAD1L1, exists in an inhibitory neuron regulatory element. This variant alters the expression of ELFN1 specifically in SST interneurons. Since ELFN1 is involved in synapse function, particularly involving a receptor called mGluR7, which suppresses neurotransmitter release, messing with ELFN1 could disrupt how these SST neurons regulate their own output.
Impairing their ability to provide that crucial inhibition.
Potentially, yes.
Yeah.
Disrupting that fine-tuning of the excitatory-inhibitory balance in the circuit so it connects the dots. GULES for specific variant, specific cell type, SST neuron, specific gene, ELFN1, altered regulation, communication breakdown, potential PTSD mechanism.
That's an incredibly detailed chain of events, all derived from this multi-omic approach on human brain tissue. Okay, this has been a really deep dive. Let's pull back for a second. What's the big takeaway here? What makes this study a genuine breakthrough for how we understand and potentially treat PTSD?
I think the biggest thing is that paradigm shift we mentioned at the start, moving away from thinking of PTSD as this uniform, generalized problem.
Because low serotonin or something.
Right, and instead understanding it as a distributed, cell-type specific regulatory disorder. The problem isn't everywhere. It's in specific cells, involving specific genes, regulated in specific ways. It's much more granular.
And the technology enabled this new view.
Absolutely.
Yeah.
Technologically, this sets a new standard for doing this kind of single-nucleus multi-omics on a human scale. Millions of nuclei, multiple data layers. It's a benchmark. And theoretically, it moves us beyond just correlating gene lists with a disorder, which is what older bulk tissue studies often did,
Towards actual mechanisms.
towards potential cause and effect chains resolved at the cellular level in actual human brain tissue. That's really the gold standard for understanding how a disease works. We're moving from what changes to how and why it changes in specific cellular contexts.
So how could this change things for people living with PTSD? What are the implications for future treatments?
Well, this directly opens the door to precision psychiatry. If we know which specific cell types, like maybe SST interneurons, or which pathways, like neurovascular pathways involving endothelial cells, are dysregulated.
We can design treatments to target them specifically.
That's the hope. Imagine developing drugs that target, say, glial inflammation if microglia are involved, or maybe enhancing GABA signaling specifically through SST interneuron pathways, or even using neuromodulation techniques like TMS or focused ultrasound aimed much more precisely at the affected cell populations or circuits identified here.
So moving away from those broad-spectrum drugs that have lots of side effects because they hit everything.
Towards something much more targeted, almost like a cellular surgery approach. That sounds incredibly promising. It really is. Now, there are still challenges, of course. A big one is translating findings from post-mortem tissue into things we can measure or target in living patients, finding biomarkers.
Right.
But this study provides the essential roadmap. It gives future research concrete, cell-specific hypotheses to test and targets to pursue. It pushes us much closer to developing truly personalized and more effective treatments for PTSD.
It's amazing how looking at things cell by cell can reveal such complexity. This deep dive really highlights that the brain's mysteries often unfold not in broad strokes, but in this incredibly intricate dance between individual cells and their specific genetic instructions. So as you go about your day, here's something to think about. If a disorder as complex as PTSD has roots in these precise cellular conversations, these specific vulnerabilities, what other conditions, maybe ones we still really struggle to treat, might also be waiting for their single-cell story to be told? Could understanding specific cellular problems unlock treatments for other neurological or psychiatric mysteries? We hope this deep dive gave you some aha moments and a shortcut to understanding this really important research. Thanks for joining us. Until next time, keep exploring.